Welcome to the Neural Implant Podcast, where we talk with the people behind the current events and breakthroughs in brain implants in an understandable way, helping bring together various fields involved in neuroprosthetics. Here is your host, Ladan.
Hello, everyone, and welcome to the Neural Implant Podcast. Today's guest is Josh Siegle, and he is actually at the Allen Institute. And I'm really excited to have him on because he's the first person from the Allen Institute to come on the show. And basically, they are a nonprofit working in brain science and both basic science and a little bit more applied science. I guess your specialty is a Neuropixels device, Neuropixels being able to read many neurons, neuronal activity from one device, thousands of neurons at the same time. Yeah, pleasure to have you on the show, Josh. Do you want to introduce yourself better than I just did?
Sure. Yeah, my name is Josh Siegle. I'm a senior scientist at the Allen Institute for Neural Dynamics in Seattle. I've been at the Allen Institute for about 7 years now. Before that, I did my PhD in systems neuroscience at MIT. And yeah, I wear a few different hats that I think would be interesting to podcast listeners. First and foremost, I consider myself a scientist. I got into this field in order to understand how the brain works at a fundamental level. And so I'm interested in questions such as how different brain regions communicate with each other and how different cell types across the brain implement different functions that contribute to behavior. But in the course of doing science, I found that the engineering side of things is also really critical. We are so far away from being able to record all of the activity that we'd like to in any given brain. As said in the introduction, we are using Neuropixels probes to record from thousands of neurons simultaneously, but that is thousands out of 70 million in the mouse brain and something like 100 billion in the human brain. And we're really quite a ways away from being able to record the activity of all the neurons in the brain. That said, with recently developed technology such as Neuropixels, I think we're starting to gain a much clearer picture of what's going on. Being able to record even thousands of neurons gives us great insights into how the brain is executing various complex behavioral tasks. And yeah, I guess the second hat would be a hardware developer. I'm not so much doing the design of the electronics, but working with engineers at a place called IMEC to develop the specifications for these probes and test them out in real living brains. And yeah, for the last 7 years, I've been at the Allen Institute. A large portion of my work has been doing the development and testing of Neuropixels probes and then applying them to the specific scientific questions that we're interested in. And then I guess the third hat is as a software developer and promoter of open source tools. So when I was in grad school, I founded a nonprofit called Open Ephys, along with another graduate student in my class, Jacob Vogt. And Open Ephys promotes the development and distribution of open source tools for neuroscience research and has really helped build awareness of the need for open source tools and helped bring open source tools into hundreds of labs around the world. So I guess those are the three aspects of my work that I'd be happy to talk more about.
Yeah, that's really cool. It seems like you've been involved in a lot of things, definite wunderkind, and now I see why MIT probably wanted to have you. But yeah, I definitely agree that I would say all science is very much limited by the tools that scientists can use, and really that science progresses with the improvement of tools. So you can think about fMRI and how much that's opened up neurology and everything like this, or optogenetics and what that's done to the field as well. Yeah, definitely. I think having better tools is, again, my biased role as an engineer. I think it's very key to this. So what is the Neuropixels probe and how does it work? Why is it important?
Yeah, Neuropixels is a CMOS electrode, which means it uses CMOS fabrication technology to put down multiple layers of silicon that contain the electrodes, as well as the hardware for filtering, amplification, and digitization of the signals detected by those electrodes. So for those who don't know, CMOS technology is one of the most widely used technologies in the world. It's what is used to fabricate all of the chips in our computers and the sensors in our cameras, but it hasn't been applied for devices specific for electrophysiology in very many cases in the past, just because of the cost required to fabricate a new CMOS device. And each fab run can cost up to millions of dollars. And it really requires a concerted effort and source of funding that's willing to place a bet that these probes will work. And yeah, basically, you can think of the Neuropixels probe as like a very long, thin camera sensor that you insert into the brain. But instead of being responsive to light, each of the pixels is responsive to voltage. And so when you stick these electrodes, this electrode array, which each probe has around 1,000 contacts, and it can read out the signals from 384 at any given time. And so when you stick these contacts really close to neurons and they fire an action potential, that creates a change in the extracellular current that can be detected by these electrodes. And because we have these electrodes are so densely packed together, each one can detect signals from a dozen or so neurons in its vicinity. And then you can get very detailed pictures of the neuron, what we call the neuron's waveform, which is the pattern of current sources and sinks that it elicits when it fires an action potential. And so we use these probes to listen in to the action potentials, which are the outputs, the output signals that these neurons send to all of the neurons that they're connected to. And we can eavesdrop on this activity using Neuropixels. And so each probe is about 10 millimeters long, which is actually longer than the mouse brain is tall. And we can stick the electrodes into almost any region of the mouse brain and listen to the activity of the neurons locally. And then we can also stick in multiple probes because they're so long and thin. We can insert. Right now, we're inserting up to 6 probes at a time. Our goal is to get that up to about a dozen probes at a time. And so we can listen to activity across multiple brain regions simultaneously, which I think is really the critical thing that has allowed Neuropixels to leapfrog the competition in terms of the quality and quantity of the data that it can produce.
Yeah, definitely. And if you're thinking about it, like you could be off by a quarter of a millimeter or something like this when you're injecting it, but basically you'd be able to offset that in software and just basically the pixel shift, move it to the side. But yeah, definitely the CMOS technology is very expensive. You can almost think of it like a stamping machine, all the semiconductor fabrication, and the stamps cost a lot, but then the actual stamping and everything like this can be very cheap. If you need to make 1 million copies of it, it's not too much more expensive than if you needed to make 10 copies. You got to have a large volume of scale. So I'm curious about this work with IMEC. I've heard about them, and definitely some researchers have worked with them as well. And what benefits do they bring, and what has your work with them looked like?
Yeah, IMEC is one of the world leaders in nanoelectronic technologies. They have access to some of the best fabs in the world and are regularly pushing the boundaries of what is possible for clients like Apple and Samsung. I don't know that those are actually clients, but companies of that scale. And yeah, they also are interested in using their technology to advance science. And there's someone at IMEC, Barun Dutta, who I think initiated this collaboration with scientists, specifically with Tim Harris at Howard Hughes Medical Institute, Christoph Koch at the Allen Institute, and John O'Keefe at University College London. And together they decided that they really wanted to invest monetary and knowledge resources in creating a CMOS device specific for neuroscience research. And at the time, nobody knew that it would actually, whether or not it would actually be successful. And so there were many rounds of development. And when I first came to the Allen Institute in 2014, they had just received the very first Neuropixels prototypes, which didn't actually have any electronics on them. They were just to test the site materials. So the metal that they use for the contacts that make up the electrodes on the probe. And yeah, I basically got to work with IMEC from this very nascent state of the probes to now when they're available for sale. They've been commercialized, so now anybody can order these probes and use them in their experiments. But yeah, it's been a very fruitful process, a lot of back and forth between the engineers at IMEC and scientists at the Allen Institute and HHMI Genelia Research Campus and University College London. which were the initial tester sites for these probes. Now there are many different versions of Neuropixels under development being tested at a variety of different places. Once we realized that the Neuropixels device, which is called Neuropixels 1.0, was going to be so useful, then there were many follow-up projects that use the underlying Neuropixels technology for different applications. But yeah, I was primarily involved in the development of Neuropixels 1.0, and now I'm working on a number of different iterations, such as Neuropixels Ultra, which has 10 times higher site density. Instead of 20 micron spacing between the contacts, there's 6 micron spacing, so you can record even more detailed picture of each neuron's action potential waveform. And also working on a project called Neuropixels Opto, which integrates light guides into the shank, so you can stimulate neurons through optogenetics, so basically inserting light-sensitive ion channels into neurons and then using light emitted from the probe itself to stimulate those neurons while also simultaneously recording their activity. So very excited about both of those projects.
What was your role again? I mean, you were the one that had been doing the experiments and then been giving them recommendations, or been like, oh, okay, this is what you guys need to improve, or giving them real world experience to the people who are actually fabricating and designing it?
Yeah, exactly. So like IMEC doesn't have any ability to test the probes in living brains. They do a bunch of bench testing to make sure that the noise levels are within specifications and like the actual like physical probe is working as expected. And then they partner with scientists such as myself to actually insert the probes into brains and record from neurons and make sure that they're useful for answering interesting biological questions. And I was also involved in the development of acquisition software for Neuropixels probes. So Open Ephys GUI, which is software I started developing in grad school, is compatible with Neuropixels probes. It's one of the 2 main data acquisition software packages that are used with Neuropixels. And so I would also give them feedback on the software API and make sure that all of the hardware was working as intended.
That's really cool. Wow. Yeah. Pixel pitch that you had mentioned, that I guess we'll get to that later, but hey guys, just a quick pause to talk about our show sponsor, Ripple Neuro and Neuromed. If you're a scientist looking to get the best quality data possible in your animal and human ePhys experiments, then talk to Ripple. Get a complete solution for your neuroscience research application backed by dedicated support. Be sure to tell them you heard about it on the Neural Implant Podcast. That will really help us out. Thanks. Now back to the show. But I wanted to talk about this going down from about 20 microns to 6. And from what I understood, I think I remember hearing that the pitch between neurons is about 50 microns. And so basically you're subsampling these. So basically each pixel you'd have maybe even 8 pixels for each neuron. Is it basically like oversampling and then you just get a better reading on what's going on, or what's the advantage of doing something like that?
Yeah, so the distance between neurons is highly dependent on the area that you're recording. In some areas, they're very densely packed, and in other areas, they're much more spread out. One thing that has plagued electrophysiologists for a very long time is what's known as the dark matter problem in electrophysiology, where the number of cells that we record is almost always less than the number of cells that we actually know to be in the tissue around the electrodes. And this raises the question of whether there are neurons near the electrodes that just don't fire any action potentials and therefore we can't detect them, or whether they are firing action potentials, but their action potentials look very similar to other nearby neurons and we confuse them with those neurons, or maybe the listening radius of our electrodes is not quite as large as we would expect it to be. And they're actually we can only eavesdrop on the activity of neurons that are very close to our electrodes. So yeah, some combination of these factors or potentially other factors mean that even when we're recording from parts of the brain with very densely packed neurons, we're still not recording as many neurons as we would expect. And so one of the main issues that we want to address with these probes, these Neuropixels Ultra probes, the ones with the 6 micron site-to-site spacing, is potentially to get an answer to this dark matter problem and say, okay, we know that there's no way that neurons are getting lost between these contacts because the cell bodies are just too big to not have the action potential be detected if they are in the vicinity of our electrodes. So looking to see how many more neurons we record with Neuropixels Ultra versus either an actual Neuropixels 1.0 probe or a simulated version of it that we get by averaging nearby Neuropixels Ultra contacts together. And yeah, so far, it seems clear that the yield of Neuropixels Ultra is a bit higher than Neuropixels 1.0 in the cortex, which is the area that we're doing most of our recordings in. It seems like neurons can get lost between the contacts of Neuropixels 1.0, especially the cortex is composed of both excitatory cells and inhibitory cells. The excitatory cells are these pyramidal cells, which have large dendritic arbors that extend throughout almost all of the layers of cortex, whereas the inhibitory cells have much smaller and tend to have axons and dendrites that are more local. And we think that with Neuropixels Ultra, we're able to detect more of these inhibitory neurons with small cell bodies, which may explain why the yield of Neuropixels 1.0 wasn't as high as we expected.
Maybe I've heard this a lot, having the brain be like a stadium full of people and you're listening in with microphones, and maybe it'd be like the shy ones or the introverts. And finally, we're able to listen to what the shy introverts have to say, even they might comprise of over half of the stadium or something like this. Maybe there's just a few of them yelling, few people yelling, and that's just, we assume that's everybody, but it's actually not. Yeah, this is really exciting. I really like this. And I guess oversampling, it really, as a scientist, it must be very exciting to prove that this is actually what's going on. So I want to talk about the Allen Institute. Do you want to talk about the history? It was founded by Paul Allen, right? One of the co-founders of Microsoft, and it's nonprofit. So what does that mean? And what's it look like? What's day-to-day look like? Is it just all scientists? How big is it? I'm very curious because I've never been there. And so I want to learn more about it.
Yeah. So I think the Allen Institute now has about 500 employees, which are spread out over a number of sub-institutes. So there's the Allen Institute for Brain Science, which was the original Allen Institute, which developed the mouse brain atlas, which is a resource that's used by almost everyone who studies the mouse brain. It's really had a tremendous impact on the field. And so given the impact of that initial brain atlas, the Allen Institute started developing additional brain atlases, such as for non-human primates and humans, and the developing mouse brain. And then, it expanded into the realm of in vivo physiology. And that was around the time that I joined, when the Allen Institute moved from mapping dead brains to mapping activity in living brains. That was what I was really excited about. So the Allen Institute for Brain Science expanded quite a bit. And then there were additional institutes for cell science and immunology. And most recently, the Allen Institute for Neural Dynamics, which I'm now a part of, which just launched earlier this year. And the day-to-day, I think it's similar to working in academic science. We're really focused on answering fundamental questions. So we do a lot of experiments, a lot of data analysis, a lot of writing papers. But I think the main difference is that there's definitely more structure there, and there is more support in terms of the project management and engineering and all of the auxiliary things that we have access to, which allow us to focus more on the science. And another big advantage of working at the Allen Institute is that we have very generous internal funding from Paul Allen. And so although we do apply for grants when it makes sense, for most people at the Allen Institute, getting grant funding is not an existential threat to their research. We're able to do a lot just with the internal funding, which allows us to engage in kind of more long-term, ambitious research, I think. It allows us to do things like the Allen Brain Observatory, which was the main project I was involved in when I was in the Allen Institute for Brain Science, which is like a large-scale, highly standardized pipeline for in vivo physiology experiments, either using 2-photon imaging or Neuropixels probes. And that was something that I think is having a really big impact on the field that would have been pretty much impossible to do in an academic research setting.
Yeah, that's pretty exciting. And 500 people, that's actually bigger than I thought. But I have heard about things like Allen Institute being almost like a paradise for, I guess, scientists, hardcore scientists, nerds, where a lot of people are disillusioned going into academia because they find out like, oh, it's actually not at all like the PhD life and postdoc life. I actually have to be writing grants a lot. And that's the thing that's taking up most of my time. And that it sounds like almost this seems, again, like a heaven for the people that are very much interested in the science, which you said you were at the beginning. So is it mostly people like you that go to this kind of stuff, or is it a mix of people, or is it, I don't know, how would you describe the people there?
Yeah, so I don't know the exact fraction that is PhD level scientists or like people who have received PhDs, but there's also a huge diversity of other staff at the Allen Institute who are really critical to its functioning. Have a large number of research associates, usually who have bachelor's or master's degrees, who are really essential to the day-to-day functioning of the Institute. We've got lots of engineers, we've got project managers, we've got the whole administrative and HR team, we've got a legal team. There are definitely a lot of people coming out of academia who are engaging in the scientific research, but there's also a wide array of people who are really critical to the functioning of the Institute.
Yeah, that's really cool. I hope I can visit one day. I'd love to see this. Is very. And then what's the point of the Institute, or what's the stated aims of the Institute again?
Oh, there's a official mission statement, which I don't have memorized.
Yeah,
it's basically like understanding the human body in health and disease, and using big open tools and team science to advance our understanding of the basic biology of the human body.
Okay. Yeah, very cool. Yeah, and so it's just talking about open source, open data, and everything like this. And I want to talk about what you had mentioned before, the Open Ephys project that you had started before you started there at the Allen Institute. Do you want to talk a little bit about this? It looks really interesting. I'm looking at the website now, and yeah, it looks good. But do you want to explain it?
Sure. So I think Open Ephys grew out of a general frustration with the state of tool development in neuroscience, where there's a lot of development happening inside every lab, but it wasn't really being shared broadly. And if you published a methods paper, it would describe the breakthrough, but there wouldn't be links to any designs or code that was necessary to replicate it. And then, there was also a lot of companies that were taking advances that were happening inside academic labs and commercializing them, which is definitely important for getting the tools into the hands of a large number of people, but they were taking these innovations and making them part of closed ecosystems, which meant that other people couldn't make their own contributions, and these innovations were then siloed inside each of these companies' platforms. And we wanted to create a platform for specifically focused on electrophysiology data acquisition, but I think this general principle applies to kind of any area of neuroscience research that you're doing, where it was really designed from the ground up to encourage people to contribute back and to make sure that it was like both easy and fun to use, but also had a modular architecture so that you could basically, like, if a new technology came along, you could swap that out with whatever technology we're currently using, and also have software that was modular, and it made it easy to share innovations because you don't necessarily have to install a new version of the software. You can just share a plugin that you've developed with another lab, and then they can immediately replicate the same signal chain that you were using. And Open Ephys, I think there are two things that really inspired and made possible the development of the initial data acquisition system that we developed. One of these was Intan chips, which you may have heard about. So there's this company called Intan Technologies based in Los Angeles that makes integrated circuits for basically amplifying and digitizing electrophysiological signals from the brain. And this kind of takes what used to require an entire rack of hardware and shrinks it down to smaller than the size of a penny. And you can stick this chip on the mouse's head, or whatever model organism you're recording from, and then you can read out the electrophysiological data with a much smaller form factor and with much less cost than was required previously. So we basically designed a data acquisition system around these chips, which at the time was considered a little bit radical, but now these chips are used in basically every single commercial data acquisition system. I think we're definitely on the right track there. And then the other thing was with the software was really inspired by tools for music production, digital audio workstations like Ableton Live that give you this modular user-defined architecture for building a signal chain. And I think is really the reason why you can do so many creative things with this software, with these kinds of software, is that they don't put any limits on kind of what is possible to do within the software, because you can infinitely reconfigure the signal chain and you can make sounds that you never thought possible using a combination of all these modules that either ship with the software or are things that other people have developed to work with it. And yeah, we wanted to do something similar for neuroscience, where the signal chain wasn't predefined. You could basically configure it to be whatever you needed to use for your experiment, have it be compatible with any type of hardware that you want to use. If you are doing experiments with one type of data acquisition hardware, such as the Open Ephys acquisition board, which is the original acquisition system we developed around these Intan chips, or you could use the exact same signal chain with a different data source, such as Neuropixels, just by deleting your data source node and replacing it with something else. And we actually based the software around a library specifically developed for audio processing called JUICE, JUCE. And yeah, that took care of kind of all the heavy lifting and making sure that all of the signals were processed in a timely fashion, and allowed us to build these reconfigurable signal chains without having to do much development work ourselves. And so, yeah, now basically anyone can build plugins for Open Ephys, and those can easily be shared with anyone else who has the software installed.
Yeah, that's really cool. And I'm always surprised at how similar basically audio engineering and obviously with these episodes, edit, remaster them, all this kind of stuff. And it looks very much similar to ePhys data, neuronal data. And so I think it's amazing. I think this is really cool because it's a good way to, like you said, standardize the data outputs and everything like this, and then be able to get it all in one place and not be siloed into proprietary company data sets or something like this. And so even potentially somebody who's not affiliated with the lab could potentially run some experiments, and maybe even they could come up with something that's interesting. And then you guys have training as well. What's the in-depth, how in-depth is this training?
Yeah, so the training sessions can be customized for whatever level of knowledge you have. And we realized that it became much easier to acquire open source tools than it was to learn how to use them. And if you didn't have another lab down the hall that was already well versed in electrophysiology, it can be quite difficult to kind of figure out how to hook everything together, and especially how to deal with the biological variation and deal with the noise problems that arise in different scenarios, and how to wire everything up on the rig so you can get the cleanest possible recordings. And yeah, this is something that we've started very recently. This was spearheaded by Alex Layton, who works at Open Ephys production site in Lisbon. And so now not only can you buy the hardware on the Open Ephys web store, you can also buy the training to go along with it, and have someone guide you through the steps required to set it up and get good recordings. And
so wait, that's like an in-person thing or like a Zoom meeting, or is it like
Right now we're just doing it over Zoom, kind of one-to-one Zoom training session. I think at some point in the future, when travel becomes a bit easier, I think we will try to do in-person trainings as well.
Oh, wow. Okay. So very high touch kind of thing. That's interesting. Yeah, very cool. And then you guys have a GitHub so people can share codes and everything like this. So I think that's really cool. You can just drag and drop and then put in your own, spin on things, but more or less it's basically building off of other people, standing on the shoulders of giants. What's been the reception of this? I don't know, the academic world liked it, have businesses liked it. A sponsor of the podcast is Ripple Neuroscience, and they're maybe one of these companies that might not be happy about siloing it, or are they happy that more people are getting into it? Have you heard from these guys?
Yeah, I think some companies are directly taking advantage of it. There are a few companies that use Open Ephys software as their data acquisition software that they ship to their customers. We've also heard about a few neural what's the word? Yeah, neuroprosthetic companies that are using Open Ephys to test out their devices. Probably the most famous one being Neuralink, which when they had a launch video a few years ago, and there's some shots that clearly showed them using Open Ephys to acquire their data. Yeah, I think many people have been able to take advantage of this already. We've also seen that most of the companies in this space now offer lower cost data acquisition systems, not quite as low cost as the ones that we sell, but definitely much lower cost and more compact than the ones that they sold previously. So I think the fact that Open Ephys was successful showed them that there was demand for kind of a lower cost, more portable data acquisition system, and so that they started offering that to their customers as well.
Okay. Yeah, that's really cool. You started a revolution, I guess, and definitely helped bring some things along. That's really exciting stuff. Yeah, this is really cool. I definitely have had a large imprint on the community. I guess as we're finishing up, is there anything that we didn't talk about that you wanted to mention?
I guess I can talk a little bit about our plans at the Allen Institute for Neural Dynamics. So this is a brand new division of the Allen Institute, which is being led by Karl Svoboda. He was a researcher at Geneva Research Campus over in the DC area, but he recently moved to the Allen Institute to lead this new division. And yeah, in my previous work at the Allen Institute for Brain Science, we were heavily focused on the visual system and kind of understanding visual perception at the level of individual neurons and groups of neurons. But now in the Institute for Neural Dynamics, we are expanding our inquiry across the entire brain, and basically no region of the brain is off limits. And we want to leverage Neuropixels technology to be able to record from many brain regions simultaneously, and specifically connected brain regions. So we can see how neurons are interacting across the entire brain and kind of decode the communication protocol that's used to transmit information between different areas. And yeah, that's just one segment of the Institute for Neural Dynamics. There's also people working on new technologies for in vivo imaging, as well as anatomical methods for tracing the axons of neurons throughout the whole brain. Yeah, I'm very excited to be part of this new team, and I think we've got a lot of exciting stuff in store.
Yeah, that's really cool. It seems like you guys are maybe expanding or getting new talent and definitely going to be taking things in a new direction. That's pretty exciting. I'm going to keep my ears open for what comes out of the Allen Institute.
Yeah, definitely.
Cool. Josh, this has been excellent. I found this very interesting and I found this informative. Like I said, I hadn't really talked to anybody about the mechanism and how the Allen Institute works. So this has been exciting. And I think your work with Open Ephys and other things have been really good. Yeah, thank you so much for coming on.
It was a pleasure to talk to you.
Hope you enjoyed the show and were able to learn something new, bringing together different fields in novel ways. Until next time on the Neural Implant Podcast.